Mahalo makana. Aloha everybody. I'm going to give our wonderful tech guy tonight a minute to just get set up so that we can, oh, okay, never mind, he's done. I'm behind. Aloha everybody, welcome to the wonderful Sphere Theater in the Maui Ocean Center. I hope you all have had a chance to see the amazing movie that happens every day in here. My name is Sarah, and I am with Maui Nui Marine Resource Council, and we're very lucky tonight to have a very special speaker as part of our monthly Know Your Ocean speaker series. Usually this is virtual, it's usually online via Zoom, but because we had a live person here on Maui all the way from the East Coast, we thought this would be a great opportunity, and she is partnered with the Maui Ocean Center. We thought it would be a great opportunity to just get the band back together and give people the chance to hear somebody speak live. So I just have a few slides that I'm going to go through. Tonight we are very lucky to have Nadège Aoki of MIT and Woods Hole Oceanographic Institution. Her talk is titled Sonic Youth, Using Healthy Reef Sounds to Call Baby Corals Home. And tonight she'll be sharing her work showing that settlement rates of Caribbean coral species can be increased by replaying healthy reef sounds on degraded reefs using underwater speakers. So we would also like to mahalo the County of Maui. They do partner with us to help support this series. So thank you to anybody watching from the County of Maui. Mahalo for your support. And if you don't know about our organization, we are the Maui Nui Marine Resource Council, and we work for clean ocean water, healthy coral reefs, and abundant native fish. And we've been doing this since 2007, and we believe that healthy coral reefs mean healthy communities. And so I was just talking to Nadège and telling her how, everything, now that I work in conservation, even more so I realize how everything is connected. And so for our organization, we do work for the reefs in a number of ways, and that means testing water quality at 39 locations in Maui. We regularly perform dives to monitor local reef and fish populations. And recently, our newest program is providing training to landscaping professionals to use natural alternatives to herbicides and pesticides. And we're very excited to put their big faces up here because this is our first graduating class of landscape professionals, property managers that took our course, excuse me, and now are able to use natural products and practices that are both better for the plant life and soil health, and that also extends into better for our reefs when we have runoff or groundwater. And so this is something that we just finished in May, was our first class, and our next in-person class is coming in October of this year. So we just want to mahalo these individuals for taking the time to learn how to do this. This presentation is being recorded, like I mentioned. Mahalo to the Maui Ocean Center crew for doing this recording for us. We're not live streaming tonight, but a recording of the talk will be uploaded to our YouTube channel as usual. And if you're on our email mailing list, you will get a notification when this recording is available. So if you have to leave or if you want to share it, you'll be able to do that afterwards. We will also have 10 minutes as usual of questions at the end, so please hold your questions until the presentation is over, and I will be helping to run the mic around so that we can record the questions that are being asked as well. Mahalo. All right, so for today's speaker, Nadège is currently completing her PhD in biological oceanography at the Woods Hole Oceanographic Institution and at Massachusetts Institute of Technology. She holds a bachelor's degree in biology from Cornell University and previously worked for the National Science Foundation and studied deep sea, oh gosh, I'm going to slaughter this word, polychaete? Forgive me please, worms at the Smithsonian National Museum of Natural History. Her dissertation research focuses on understanding the ecological role of underwater sound on coral reefs, particularly the impact of sound cues on early life stages of corals and developing methods for using soundscape recordings to monitor reef health. So she's very impressive. I'm already intimidated, but can we please give a warm welcome and aloha to Nadège, please?
Aloha, everyone. Is this on? No.
Now it's on. Okay. Hi, as Sarah said, my name is Nadège Aoki. Some of you, if you were here earlier this week for the coral spawning night here at Maui Ocean Center, you may recognize my face. You may even recognize a couple of my slides, but welcome back, and hopefully you'll learn a little bit more about the work that we're doing here with Maui Ocean Center. I'm really privileged and lucky to be able to be here in person, and like Sarah said, this was a really serendipitous timing of our research trip overlapping with the speaker series that you guys have here. So I'm here representing work that is a collaboration of many different people. So the two sort of principal investigators on this project, Dr. Erin Mooney and Dr. Amy April. Dr. Mooney is actually here in the audience tonight. Dr. April is also is back at the Woods Hole Oceanographic Institution in Cape Cod, and just so you guys know, you can look up both of their labs and learn some more about the awesome science that we do. I also would like to just put in a plug for the Reef Solutions group at Woods Hole. So at Woods Hole Oceanographic, we do all kinds of world-class pioneering oceanographic research on every topic you can think of in the ocean. And recently, a group of scientists at WHOI, what we call it, at Woods Hole, have come together to form a group that we call Reef Solutions, and it's basically interdisciplinary science trying to think of innovative solutions for monitoring and restoring coral reefs. So please take a look or click if you have time. And I just want to also kind of pause on that note and think about partnerships and collaboration in reef science, and just acknowledge the many, many people who have helped with this project, with all of the reef work that we've done. For coral reef science particularly, it's really impossible to do it without the help of the local population who know the reefs, who snorkel them, who dive them every day, and who understand what the issues are and where the science needs to be directed. And also, in our case, we just really appreciate having these collaborations help spur us to better science. So most of the work that I will be presenting today is actually work that was conducted in the U.S. Virgin Islands, a couple projects in collaboration with the National Park Service, the Nature Conservancy there, as well as CORE Virgin Islands, which is a volunteer group which does a lot of monitoring and things like lionfish removal in the islands. And here on Maui, we're really excited that this summer is the beginning of an at least 2-year partnership with all of the partners listed here: the Maui Ocean Center, Division of Aquatic Resources, the Hawaiian Islands Humpback Whale National Marine Sanctuary, the Nature Conservancy, and of course us from Woods Hole. And it's just really, really important to us to highlight those partnerships and to acknowledge that this is, while I'm the one speaking, I'm representing a large and collaborative group of people. So, just putting that out there first. So today I'm talking about sound in the ocean. And many of us, maybe, when we're in the ocean, we're not really thinking about what we're hearing necessarily. But if you are willing to open your ears a little bit to what the ocean has to say to you, you'll notice that there is tons of sound going on in the ocean. So sound actually travels 5 times faster in the ocean or in water than it does in air, and that's because the ocean and water is a much denser medium than air is. So the particles in the water are closer together. That means they can transmit those sound waves faster. Also, things like light attenuate very quickly underwater. So if you are a marine animal, it's likely going to be more effective for you to develop ways of using sound, either producing sound, sensing it, to communicate and sense the world around you, compared to using something like vision, which will not be that useful if all the light is being absorbed 10 feet away from your eyes, or at night when it's pitch black in the ocean and you can't see. So lots and lots of marine animals are adapted to use sound underwater. We are all probably very familiar with marine mammals, whale songs, and those kinds of things, but fish and invertebrates are also very common users and producers of sound underwater. And we can study this sound as scientists. So there's a whole field of marine science that we call acoustic ecology, or sometimes soundscape ecology, because you're studying the entire landscape or soundscape of the acoustic environment. We use tools called hydrophones instead of microphones because they work underwater. And by recording the ocean, we can remotely monitor areas that we may not be able to access with much frequency with divers, or that are just too remote for us to regularly survey. And we can get an idea of what kind of animals are there, what sort of biological or human activity is going on, because you can obviously record not only animals, but also boats and things like that. And we can also, by recording those sounds and sometimes playing them back in controlled environments, we can study how animals respond to sound and what their sensory capacities are and what the importance of sound is to animals underwater. So what does this actually sound like? Again, if you were here earlier in the week, I'm sorry, this is a repeat, but it's a pretty cool clip. So what you see here is an image that we call a spectrogram, and that is a way of visualizing sound and being able to look at it through different frequencies. So from left to right here, on our x-axis, we have time. This is showing a 30-second audio clip. On the y-axis, we have frequency. And the color, so the brighter white or yellow or orange, is showing higher amplitude or louder, more intense sounds. So as I play this clip, you should be able to hear different sounds, and some of these are actually, you wouldn't expect it, but they are produced by fish. Some of them are produced by shrimp on the reef. So there's an entire group of shrimp called snapping shrimp that have a really big claw that they use to snap really, really fast. It sends a little jet of water that they use to stun prey on the reef. And that snap produces some of the loudest biological sounds that are recorded underwater. So I'm just going to play this and keep your ears open and see if you can follow along. Yeah, the sort of grunts that you heard were the fish, the swooping sound was a humpback whale, and then all that crackling in the background is actually shrimp. So that's just an example of something that you can hear by listening. And you can probably, you'll hear snapping shrimp on any reef here if you're snorkeling, diving. And so we can record these sounds, right? And we can look at the patterns in them and see what they tell us about animal activity. So this is sort of a similar kind of representation of audio here, but blown out into another axis so that you can look at how it's changing over the course of a day. So those peaks that you see, the big red peaks here, are actually choruses of fish. So many fish exhibit what we call this crepuscular activity, where they make more noise, they chorus together at dawn and dusk. And that is, a really very useful indicator that we can use to say whether or not, a reef has a nice healthy fish community. If we're seeing that kind of behavior, it tells us something also about when the animals are active, when they're not. So we can start, by recording these soundscapes through space and through time, we can compare how different reefs sound from one another and how that reflects the habitat quality. And when we do that, we start seeing these patterns where things like coral cover, which is one metric that we use for indications of healthy reef, tend to be associated with higher sound levels, particularly in these low frequencies that indicate fish calling activity. And what we also notice is that degraded reefs without a lot of fish, without a lot of coral cover, they lack a lot of those healthy biological sounds and signals. And these relationships are not precise, it's not one-to-one, because the contributions to sound are not always, an indication of biological health. But in a lot of cases, you see this general relationship, and you can pretty broadly use the soundscape to tell you something about how a reef is doing. And that's a really great kind of tool that we can use for monitoring. It's a way, there's a whole technique called passive acoustic monitoring, where you can passively record the ecosystem and then look at that recording and tell you something about the ecology, right? But it's not just a monitoring tool, this is also an ecologically significant sensory input, right? So we know from the last couple of decades of research that many reef animals are responsive to this sound as well. So it's been demonstrated that the larval stages of a broad range of families of different reef fishes, as well as some invertebrates like oysters, are attracted to these reef sounds. So this graph I'm showing here is from an experiment, paper that was done on oyster larvae. And basically here on the left, you've got kind of their setup where they had a speaker, and then they had some of these larvae in cultures that they exposed to different sound treatments or no sound. And they saw that a lot more of these larvae settled. And by settling, I mean they came down and attached to the substrate. So when we think about benthic or animals that live on the bottom or that don't really move much as adults, a lot of them have a mobile stage when they're juveniles, when they can swim around, and then at some point they settle and they decide to drop out of the water column and pick their home. And that process is something here that was significantly triggered by the addition of that sound cue. So this has got us thinking about like, hey, what about coral larvae, right? Corals are such fundamental, important animals in our reef ecosystems. How is sound influencing them in their mobile larval stage? And a paper about 15 years ago at this point demonstrated sort of the first possibility that corals could respond to sound. So they basically had a big PVC tube, you can kind of see it in the gray coming out of that speaker, and they put a bunch of coral larvae in it. They had a speaker on one end, and then they had some people observing for the next, I forget how long, hour, a couple of hours, where the larvae decided to congregate. And they all, or not all, but a good proportion of them, right, all kind of clustered towards that sound source, toward that speaker. So that tells you that these larvae are sensitive to sound, or they're able to exhibit what we call phonotaxis, so it's movement towards the sound. And then our lab kind of followed up on this and thought about, okay, how are the natural sound cues in the environment affecting settlement? So in this case, we found 3 different sites, so a sandy site, a more degraded reef, more healthy reef, all of which kind of follow the gradient of low to high overall sound levels, put larvae out there, and observed that their settlement rates followed that same gradient. So low sound was less settlement, here in the gray, kind of a little bit of ambient sound, had a little bit of settlement in the blue, and the higher, healthier reef in the red was much higher. And like, why do we care about this, right? So the larval settlement is a process that is incredibly important for coral reef populations. So on the left here you have sort of the life cycle of a coral, right? And when corals are adult animals, they're attached to the reef. They can't move, they can't, if it starts being polluted, if the reef is fished out and they're getting overgrown by algae, they can't go somewhere else. So the only time when they're able to migrate or colonize new spaces is when they're in their mobile larval stage. And for many species, if they don't encounter kind of the right cues for suitable habitat during that stage, they may just die in the water column and perish. So it's a really important process that we are also observing is declining globally. So as coral reefs decline, as the sort of overall coverage of corals that we have declines, and as our reefs deteriorate in their condition, they don't have as many of those kind of chemical and acoustic and other sort of nice signals for the larvae to latch onto and to find a good place to live. And just so you can tell what you're looking at here, the little video on top is swimming coral larvae in that searching stage for where they're going to live. At the bottom, it's once they settle, they metamorphosize and turn into what we think of more traditionally as a coral polyp. And that, figuring out what is triggering them and what are they looking for during that mobile stage, and how can we potentially help them along to their new homes is a big question. Right? That leads us to the idea of, okay, if they're responsive to sound, can we just play them sound? Can I just like drop, you know, my like waterproof speaker on a reef and play some Beyonce or something, and that'll just like bring all the corals in, and that'll save the coral reefs, right? So that's not really how it works. And there's obviously there's so many questions that you have to establish first, kind of scientific sort of basic principles that you have to figure out. Like, how are you going to play sound underwater, or how are you going to make it actually sound realistic to what a natural reef sounds like? Are the larvae going to respond? What do they actually care about? Like, what frequencies, what sounds are they listening for? Are they actually able to sense anything kind of more specific than just sort of an overall level of ambient vibration? Is this going to have an impact on other organisms? Or at the same time you're putting a speaker in and attracting coral larvae in, are you also attracting their predators? And it's sort of not actually doing anything to the system. And does this actually, will this make an impact in terms of the other negative cues and other forcings that we're seeing on the reefs? So these are like all the big questions that are out there that could make up many, many PhD theses. But I've just been focusing on a few of them for mine. And starting off with sort of the fundamental question of like, all right, to test some of these questions, you're going to need a system that's going to work, that's going to allow you to sort of play realistic sound underwater. And you might think that's easy, but it's actually kind of a complicated question of how you do that, especially on reefs that are maybe remote or deeper. So at Woods Hole Oceanographic, we are pretty good. We have a lot of engineers on our team in Woods Hole, and they are really good at building stuff like this. So we created what we call the Reef Acoustic Playback System, or the RAPS, which it says here is a robust, customizable, solar-powered underwater speaker system made generally with off-the-shelf components. So we're trying to keep the cost down, trying to make it something that institutions around the world could replicate and manufacture. And this is what it looks like. So we'll basically put a speaker on the bottom. We'll have our buoy at the top with a solar panel that's charging the system and that's powering it. And this is just a quick little look at what it might look just putting that buoy in the water. You can see it looks a little bit like, you know, a mooring or something in one of the videos. But yeah, this is a beautiful, beautiful island of St. John where I've gotten to spend some time. It's a hard life. So after we've developed the system, right, we can use it to test, okay, are coral larvae going to settle in response to replayed reef sounds? The study that I'll be talking about took place in St. John, in the U.S. Virgin Islands. We used a very common coral there called Parietes asteroides. It's also called the mustard hill coral because it's mustard colored and makes little hills. We were able to collect some of the larvae from this coral. We placed them in enclosed settlement chambers, exposed them to enriched sound and no sound treatments at both healthy and degraded reef sites. So these are some of our little chambers. So it's basically like a specimen collection cup, and you put in a little, we call these are like little ceramic stilts that are used in the pottery field, but we make use of them as little attachment sites for coral larvae. We have our speaker, and then in the end, we assess where did the coral decide to settle. So did they do it in the cups that were on a site enriched with sound, or on one that wasn't? And we actually ended up having 3 sites. One of them is one where we played sound, and the other 2 we ended up not playing sound for various reasons. We looked at settlement and how it changed as we got farther away from the speaker as well. And what we were playing back to the corals were sounds recorded from a local reef in the area about like 10 years ago. So when the reef, it's a reef that we know is like more fishy, more corally than the others, and also is 10 years ago was probably doing a little bit better than it is now. So this is a little video of the setup. So you have a speaker here. These are some of our little cups where we're putting corals in. That's me and Erin having fun underwater. And that's kind of like a tree structure in the middle. So these are some of our acoustic recording devices, our hydrophones, that we put out during the experiment to monitor what the sound actually is that the corals are hearing. And so what did we find? We found that there was significantly more settlement, more of what we wanted to see. On average, it was around 2 times as much, and if you compare sort of our lowest scoring cups with our highest scoring cups in the sound treatments, it's 7 times as much. And yeah, so these 2 graphs that I have on the left and right, we repeated the same experiment twice, once in June and once in July of the same year. From left, on the far left, the red dots, the red box plot is showing data from our sound enriched site, and the other 2 are essentially controls. And you can see that the red is higher than the blue or the gray. And on the y-axis here, it's basically the percentage of larvae in the cups that settle. So anywhere between somewhere around like 10% to 70 or 60% of the corals settled in our experiment. And this work was published earlier this year. So that was really exciting. It meant that, okay, sound is having an impact on this process. And something else that we were looking at, right, was whether or not this changed as you got farther away from the speaker. So that would indicate that there's something, if it didn't change as you got farther away from the speaker, that might indicate there was something else in the experiment, something else in the environment that was changing things, right? So what we would expect to see in our controls is the effects of basically be random, to have basically the same amount of settlement at every distance. And that is what we saw in our controls. And then in our experiment, in our enriched sound site, what we were hoping is that the effect would decrease as you got farther away. So as the sound is less intense, they're settling less. And that is pretty much what we found. There was a little bit of like funkiness going on at one of our sites, but overall we saw this decreased distance. So we saw settlement impacts decrease with distance from the speakers, and at our experimental site, they remained elevated above our controls at all distances, so up to 30 meters away. So basically what that means is that we were able to project a radius of at least 30 meters where if we play sound, corals are responding to it, right? We also looked at this a little bit through time, where if you expose corals compared to 24 hours or 48 hours or 72 hours of exposure, does that make a difference of how long they're exposed to it? Because a lot of these corals, they have different kind of amounts of time that they tend to spend in the water column, different times that their larvae are viable for settlement. And here again, basically the takeaway is that the red graphs here, the red plots are higher than our controls at every time point. So within 24 hours of exposure to this treatment, we were starting to see differences between our controls and our experiment. And there's a little bit of a kind of funky result that we got that I won't get too much into, but when we tried this with the second species and did basically the same thing, where we exposed some corals to sound treatments for 24 hours, some for 48, in that case, we got a different result, where the only time when we saw a significant increase in the sound effect, so again, the red plots here, was during the first 24 hours. And that was different from this other species we tested, right? And that's interesting and makes you think, okay, so what's different about these 2 species that we're working with? And this is, again, the results from our first species on the left, second species on the right. And the difference is that in the first 24-hour set, the red bar is a lot higher than the gray bar, and then in 48 hours, or 48 or 72, they all kind of flatten out over here, while on the left, the Porites stays much higher than the one on the right. And when you look a little bit more into the actual biology of these 2 animals, you see differences that might explain this. So the Sphadia fragum, the little coral up in the right-hand corner here, is the second species we looked at. And these larvae, when they come out, they have a very short amount of time in the water column when they are most ready to settle. So they usually settle within 36 hours of being released, so less than 2 days. And that means that they're like really eager to do it quickly, and then once they get past that 24 hours, they basically might just be kind of desperate to settle anywhere. And that, in that case, it might not really matter whether or not you play sound or not. In contrast, our other species, they have larger lipid reserves, which means they can last longer in the water column without settling, so they can be more picky, and that means that they can stay in the water column as larvae for a lot longer and really look for those settlement cues. So in summary for these results, basically what we found is that multiple Caribbean coral species settled in response to replayed reef sounds. And what's the implication there? It's exciting that maybe we can use acoustic enrichment to help restore our reefs. Maybe we can put speakers down in areas that are degraded in some way, where we're not seeing the recruitment of juvenile corals that we want to restore the area, and maybe that will actually be effective. However, the impacts are pretty variable by species, so that means that you have to consider the life history of the species that you're trying to target, and means you need to do research on different species in different regions. So that brings me to here, and why I'm here this summer. So as part of the project that we started here with Maui Ocean Center, we've been trying to expand some of this work to a variety of coral species, not just in the Caribbean, but also in the Pacific. So this summer, we've collected 5 different Hawaiian coral species, the cauliflower coral, base coral, lobe coral, finger coral, and rice coral. And these corals represent some of the most common, most abundant corals on the reef here, but also a mix of different reproductive modes and different life history strategies. So in some of these species, the larvae take a long time to develop. Some of them, they come out very, very ready to settle. So it sort of allows us to get at some of these potential differences between species. And a lot of what I have spent my time this summer doing is figuring out kind of what are the best ways to capture and rear and raise these larvae for experimental use, because it's not super easy. If you were here for my coral spawning talk, you know that we only know kind of the timing of these species by a lot of luck, honestly. A lot of divers being out on the reef at the right time and then deciding to go back at the same time the next year. Many species only spawn once or twice a year, so you have to be there at the exact right time to catch it. So on the left here we have the rice coral spawning. So if you were here at Maui Ocean Center, this is the species that you saw. What you didn't see is sort of what happens after, right? These are egg sperm bundles that the corals release that float up to the top of the water column and then break apart. So in the middle here, this image is some of those bundles beginning to break apart, and the little kind of mist you see coming out of them is the sperm releasing out as the eggs break apart and the sperm comes out. And then on the right here we have, so currently we have been rearing these embryos for the last 3 days, and they're just starting to have a little movement and have a little bit of this spinning behavior you see on the right. But these corals take at least 5 days to be ready to start swimming and start moving around and exploring the reef. And keeping them alive in the lab is a challenge, and that has been a hamper on a lot of larval research, is kind of getting those conditions right. And then once we kind of have these larvae ready to go, we've been exposing them to different acoustic conditions in the MOC nursery. So if you've ever been on a tour at MOC, they usually take you back to the coral nursery, and you might have seen us in the last couple of months there with our kind of big spread of equipment. So we have these yellow boxes here underneath the tanks. Those are the same kind of pelican boxes that we put onto our buoys to power our acoustic system, and we just have a bunch of those here in the lab, and in these like pink tubs that you see, we have a speaker in each of them. They're covered in foam to insulate them, and we've got water flowing through that system, so it allows us to have these little kind of isolated acoustic chambers. And in each one of them, we're exposing larvae of different species to different acoustic treatments and basically seeing what happens. And I can't really speak yet to what is coming out of that because these experiments are still in progress. But this is sort of, what we are looking for ideally, right? On the left here are settled corals. So these are little baby corals that have settled, they've picked somewhere to grow, and hopefully they will then survive and grow for years to decades. On the left, one of those pictures has them kind of glowing green, and that's the way that we look at them under the microscope is with green fluorescence. And then the other picture is after just a week, that coral is already starting to grow tentacles. It's already kind of ready to start living its life. But getting to that point and then getting that coral kind of through that first year or so of life is a pretty big challenge, and we are just trying to do our best to understand how that process works and how we can help them along. And next year we'll be back, also working with Maui Ocean Center and the Humpback Whale Sanctuary to do a lot of this in the field, because one thing about acoustics work is that it's never quite the same in a lab, in a tank, as it is in the ocean. And if you think about it, that makes sense. If you kind of put a speaker in a box, in a small box with a lot of rigid sides and surfaces, that sound is going to bounce around and reflect and kind of sound weird compared to if you did it out in the open space. So that also impacts our experiments, where the acoustic environment we're exposing things to in the lab is not really exactly what it would be like on the reef, and therefore we can't necessarily assume the same behaviors are going to happen. So next year we'll be back, we'll be putting some of these speaker systems out on some Maui reefs, and we're going to be seeing what kind of coral responses are. So I have so many people who have been involved in this project. You can see from this list that it takes a village, it takes an army to do this kind of science. We're very lucky to have funding from a variety of sources, including the National Science Foundation, and we obviously have to done all of this under permits from the Maui Division of Water Resources and the National Park Service in the U.S. Virgin Islands. At this point, well, happy to take questions. I had a good slide.
Yes, applause is well deserved. Thank you so much. We're going to switch the HDMI cord right now. Just a second. There it is. Okay, the moment everyone has been waiting for. How about another hat first? You can make. Oh, wow, that was wonderful. So we do have some time before we need to wrap it up for some questions. So I think myself. Oh, we have lovely Makana and Daniel as well. If anybody has any questions, can you just throw your hands up and we'll try to get the mic to you. We are recording it, so we would like to also catch the question on the mic. Okay, this gentleman right here. And then
how confident are you to the future of being able to do something constructed on a reef on large enough scale to actually make an effect?
I think what's going to have to make that possible is kind of doing this in concert with other kind of efforts that are already ongoing. And that's where I sort of see this tool fitting in is, if you just playing sound or just maybe attracting some corals to settle at a site, one place that has been degraded for some other reason, that's not necessarily going to fix the underlying causes, right? So we still need to be doing things like addressing ocean warming that's causing all of these, massive heat waves and things like that. But I think that right now what's nice about this tool is that it's pretty easy to kind of bring a speaker along to whatever other restoration project you already have ongoing and kind of add it and sort of see if it helps the process along. So I'd say this is a question kind of about coral restoration in general, is really, are we going to be able to scale it up fast enough and at a broad enough scale to preserve coral reefs? And I think the answer is degradation is happening on coral, reefs around the world, and it's unlikely they're going to stay in their present form no matter what we do. But that doesn't mean that we have to kind of give up on corals entirely or reefs entirely, right? And I think that it needs to be strategic about like where are you going to try to preserve, where are you going to try to restore, and how can you make sure that those areas are well set up to allow corals to thrive there in the future.
There was a gentleman in the orange shirt right there.
Aloha. Two questions. One, with the increased sound in the areas of greater coral cover, which organisms are, do you know which organisms are responsible for that increased sound?
So not in every case. So in a lot of those areas, a difficult thing about some of this reef acoustics is that we know kind of what broad category, like we know what our fish sounds, but we don't necessarily know which fish are making them, because determining what sound kind of goes with which animal is its own project. You have to get kind of video and audio of the animal making the sound at the same time and have confidence that they're together. But yeah, the increased sound that we see on more kind of healthier coral areas do tend to be in that sort of the lower frequency bands, kind of below 3,000 hertz or so, and that is where we mostly see fish activity, and so that's largely what we think is contributing to it. There also is sometimes more snapping shrimp activity in those areas because there's a lot more structure on the reef, and that allows more places for invertebrates to sort of hide and live. But yeah, we don't, it's very variable, both. There's a lot of heterogeneity in the data and on the reefs.
Super, thanks. And the second question was, I know it's really early new research, but in terms of the sounds you're using to broadcast, are you using like varying both volume and also, for example, which sounds like I noticed there's this increased symphony at the dawn and dusk. Have you used like just broadcasting at dawn and dusk like that, or recording those sounds?
Yeah, no, that is like kind of exactly the thought process that we're going down. You know, we kind of started out really trying to sort of replicate the natural daily cycle. So we would sort of record 24 hours of sound from a healthy reef and then kind of play it back and sort of try to match up at those times. But part of what we're doing here at Maui Ocean Center is trying to think, like, is that the best way? One thing we're doing here is we've been playing some sounds recorded in the Caribbean to these Pacific corals, and it's sort of unlikely that they're really going to be able to tell that much of a difference, but we don't know. And that actually is working pretty well so far, is that they are responding to those Caribbean sounds that have a lot of fish calling in them. So we think that there's definitely a possibility to sort of make, you know, kind of a greatest hits, like a playlist of some kind, and broadcast that. And again, the timing is an important aspect of it too, of like when in the month or the year is this actually going to be effective. That's going to be kind of thinking about those coral spawning cycles as well. But what's nice about our system is it is solar powered, and so far it kind of can play continuously. So right now our process has been kind of play sound as loud as we can and sort of see if the effect is there, and then we're going to try to sort of like modulate it a little bit more.
Any other questions? Right here in the blue shirt.
I was wondering if, I don't think it's part of your study, but if there might be an impact on the unnatural sounds that are in the water, boats in particular, and, you know, a lot of activity like that.
Yeah, that's sort of noise pollution in the ocean is kind of its own complete other subfield of research that is, you know, related and intertwined. And yeah, there's been a pretty good amount of work on reef fish, especially, about the impact that motorboats and things have on them, and that kind of chronic stress that can lead to. And it definitely does impact behaviors. In some horrible fish, it can kind of like stunt their development a little bit. But the sort of question of like, what's too much sound, like what's good versus bad sound, and sort of how, especially on reefs where, you know, you have dive boats or fishing boats kind of going by like every day, are the animals just sort of acclimatized or adapted to it. That's all kind of ongoing active areas of research, but the unnatural or the anthropogenic sound going into the ocean has a lot of impacts, and not just on reefs, but also in other things, like global shipping traffic noise and things like construction happening in the ocean to build things like platforms for offshore wind farms. I was just talking to Sarah about that. That generates a lot of noise, and some of it is extremely acute, kind of these like big strikes from hammers and things underwater, and we don't really know what the extent of those potential impacts are on marine species.
Any other questions? Before I ask my question, I have a question. My question is, so I know, this is, I don't think this was necessarily mentioned in the talk, but you're kind of coming up on this last week of wrapping up this year's, I guess, study. You mentioned coming back next year as well, and I was just curious, what does wrapping it up look like this, to close out this year, and what does the work entail when you guys return?
Yeah, so this week we're sort of wrapping up, we're doing our work with our last species. So this summer, we have coral spawning events from different species that happen sort of throughout. We've been working on them from May until now, and we just had our last spawning event go off, so we'll be sort of prepping our last round of experiments, packing up, and then next year it's really going to be trying to move this more into a more realistic scenario on the reef, looking at what are our natural rates of settlement and larval production on the reefs. Are there areas sort of like on the Maui coast here where we might expect that larvae are being brought by the currents, but they may not be settling, and see whether or not we can lure some of them out of the water column. And I'm really excited for that just because, that the fieldwork and the more realistic applications are where we can make more of an impact, right? Doing this in the lab is really valuable. It provides us a lot of really useful information because you can control so many more factors. And it also has potential relevance to things like coral nurseries like at MOC and spawning events here, and people who want to make things settle and grow in lab or tank environments. But if we want to rebuild the reefs, we need to be doing this on the reefs, right? So I'm excited about that next year.
Is there any chance for I know that you said it's about a 2-year process, so next year being the second, is there any possibility you may start a new project or extend or?
I think definitely depends on things like funding and how that shakes out. But we actually had a few years ago had an ongoing project here monitoring the sounds of different reefs around Maui, and that actually has been kind of kept up. So we have a very long time series of recordings from a bunch of the local reefs here, and so we're kind of excited to now have the opportunity to be sort of back here locally and kind of making use of some of that long-term data.
Amazing. Any last? Please, we've got time. Yep.
I would be interested in knowing what frequency ranges you guys have been using and listening to.
Yeah, so the sounds that we've been kind of broadcasting range anywhere from, I think, 10 to 20,000 hertz or so. Again, so a lot of that fish calling activity tends to fall in the pretty low frequency range. Actually, almost below 1,000 hertz or 2,000 hertz. And then a lot of where you see more of that snap and shrimp invertebrate activity is kind of more between 2,000 to 20,000 hertz. We expect, just based on other marine invertebrates, that the relevant frequency range for these larvae is likely to be very low, and they are likely not actually sensing higher frequencies and likely not also only sensing the sort of vibration from the sound. So larvae do not, other animals will have a lot of different hearing mechanisms, but one thing that's important is sort of having kind of air pockets or a swim bladder that responds to the pressure of a sound wave. And larvae, they don't have that, they potentially have these sort of like cilia or hairs on their body that may be deflected by vibration, and that may be how they're able to sense it, but their range of sensitivity is likely going to be restricted mostly to those low frequencies, which is where a lot of biological activity occurs, but also is where a lot of things like boat noise or boat motors generate a lot of low frequency noise as well. So there's sort of a lot of stuff going on down there.
What are your thoughts on applying this to new artificial reefs in areas that might be acoustic dead zones or not live?
I actually think that that's potentially a really great application of like places where, there's at least one artificial reef project going forward in the Virgin Islands that we're kind of excited to potentially partner with, because the idea, right, is that you could bring in, during a reef that you're trying to populate, you could sort of kickstart it a little bit by adding in some of these artificial cues to try to bring in some of the animals that you want, that then will hopefully set up their own, ecosystem and be self-sustaining eventually. So yeah, I am all for that, and I think that that is actually an area where this may have more relevance than others potentially.
How does amplification kind of factor into your research, especially in a situation like in the laboratory in a tank with reverberation versus open ocean?
It's something that you have to really kind of quantify. So you cannot say that in a tank that, the reverberation, the sort of reflections, that is necessarily replicating the open ocean environment. But what you can do is you can record what your levels are, you can record the pressure and particle motion and then you can report it and say like, hey, when you're at this level, this is the behavior that we observed, and take that with a grain of salt when you're expanding it to the open ocean. So the best you can do is kind of try to make conditions as realistic as possible and quantify and describe them and just make it clear what you're doing and the caveats that are associated with that.
I might have missed, did you say that you have areas in mind around the island for where you'll do the field work?
A little bit. So we've done a little bit of, part of what we've done here on this trip is sort of scoping out some sites. I think some of them are kind of like off of Oahu a little bit, but a little bit farther offshore than like people would generally swim to. But I don't think we really have it all set. So a lot of that is sort of in conversation with kind of our folks at NOAA, folks here at MOC, and sort of where they think would be good spots for us to do these experiments that are good targets for our research, but also are potentially not places where everything's going to get run over by boats or curious tourists or things like that.
Any other questions? Perfect. Okay, well, how about another round of applause for our speakers today? Thank you so much. And thank you all for being here. Mahalo to the County of Maui, as always, for the support. And our next speaker will be, actually probably more than one, but it will be Huio Kawaiola. That is the water quality organization that we work with, and they will be discussing the water quality report that they release every 2 years. So please keep an eye out for the Reef and Brief, our monthly newsletter, to stay updated on when that comes out. And like I mentioned earlier, this is being recorded, and so once we get the recording, we'll go ahead and we'll upload that to our YouTube channel. We are Maui Reefs. We're pretty much Maui Reefs on everything except for Facebook, so you should be able to find us easily that way. And we just want to mahalo our supporters and our sponsors, not just for this talk, but for everything that we do. We are a very small, local, nonprofit organization, and so we are very thankful for our generous and supportive community. We do what we do because of you. And so that's all we have for tonight, you guys. Thank you so much for being here. How about a round of applause for our Sphere staff for hosting us, for making us comfortable. So thank you so much for being with us. Please drive safely tonight as you go back to your homes. And we will see you guys next month. So thank you very much.